Well, thank you everybody for coming out today and hanging out with us. We really, really appreciate your support. And in the audience, I see a lot of former colleagues and friends and some new faces, which is very exciting too. So thank you, John, for organizing this meeting. Really appreciate it. My name's Yasmina Abdiche. I'm the Chief Scientific Officer at Carterra, and I'm really excited to tell you about our technology. And we're going to just start with why high-throughput antibody characterization. So a little about Carterra. So Carterra is not a new company. It was actually founded in 2005. Headquarters are in Salt Lake City, Utah, and it was formerly called Wasatch Microfluidics. And it was based upon licensing out a continuous flow microspotting technology, which is a really efficient way of flow printing proteins onto the surface of a slide or a gold chip. And a really excellent application of making these microarrays of proteins was to combine it with SPR. And in the early days, Wasatch Microfluidics partnered in a non-exclusive relationship with IBIS from the Netherlands. So Wasatch's flow printing and IBIS's SPR detection were used as a combo system to seed the concept of high-throughput antibody characterization. But the dream was really always to integrate these two capabilities together in the same box. And so Josh, who I'm going to just embarrass him now, who is the co-founder of Wasatch Microfluidics and Carterra, maybe just stand up so everyone can see you. He's our CEO and co-founder. He's awesome. We had been collaborating for many years while I was at Pfizer. I was there for about 12.5 years. And we were collaborating on this concept of integrating the two technologies. So I helped him to get some seed funding from Carterra towards this dream, got them $1 million, which was awesome. And then that seeded their investment and attracted a more significant investment from a well-known investment company in the city. And they were basically acquired in 2016 and rebranded as Carterra. And then at that point, I joined them as CSO, and they expanded the management team, R&D, and commercial teams. And we have 3 labs, one in Dublin here on the East Bay, one in Salt Lake City, and one in Newtown in Pennsylvania. So we're really excited to move this concept forward. And I'm going to show you the instrument and the capabilities today. We've been commercializing this, and I've already sold units in the States, one in France and one in Australia. And we were in Japan last week seeking out a distributor for Asia Pac. So we're super excited about that. So this is really the whole kind of tenet that it's based on. If you were to look in the night sky and focus on a very, very small part, which seems empty, if you were to use a very sophisticated telescope, what they actually found was that there was actually millions and millions of stars, galaxies there that you didn't even really know about before. And so this analogy is basically, once you have more sophisticated tools, you can see things that you couldn't see before. And in the context of drug discovery, this is huge. You often don't know what you want until you find it, but having more information earlier and really appreciating your antibody libraries at the earliest possibility just gives you more information to make better choices. So, as most of you know here, don't need to preach to this crowd, but for anyone who doesn't know, therapeutic antibodies are really hot in pharma, and about half of the sales are because of biologics. The other half are small molecules. And so this is like a really lucrative area for pharma. And Humira itself, it was like a profit of like $16 billion a year. So this is huge business. So anything that can accelerate the discovery of a therapeutic antibody is highly advantageous. And I've been working in the biosensor field for about 20 years, and biophysics is really agnostic of the target because it's looking at molecular level interactions. So when it comes to therapeutic antibodies or just drugs in general, understanding the mechanism of action is really key to getting it ever approved. You need to understand how your antibody works or how your drug works. And biophysical tools such as biosensors allow you to generate information about the binding and understand the mechanism of action. So when I was at Pfizer for 12.5 years, we were investing a lot of money in the generation of antibodies with using different platforms like hybridoma, transgenic animals, cloning technology for synthetic libraries. And there were so many antibodies made, but the analytical tools lagged orders of magnitude behind in their capacity. So I was super frustrated because my colleagues would have all these antibodies and I was forced to undersample their libraries. So I really couldn't appreciate the full depth of the kinetic and epitope diversity that was contained in the library. And you may actually be throwing away a blockbuster candidate. And I've noticed there's a paradigm shift in the community that people are making smarter libraries and really demanding to really understand what they've made. And if you actually look deep enough into your library, it's highly likely that you will find a clone that is near perfect and requires minimal engineering, and then you basically have a lead right there. So if you do deeper characterization earlier, you're expediting the drug discovery process. So SPR, or surface plasmon resonance, for anyone who doesn't know that acronym, it's always been thought of as a low throughput technique and kind of had a secondary position in screening. It's really been thought of as a characterization tool, not a screening tool. So the concept of high throughput SPR is basically a new paradigm, and this is actually providing a disruptive technology to the industry. If you were able, at the earliest stages, instead of just doing ELISA, to actually get very detailed information about your clones, detailed binding kinetics, affinities, understand the epitope landscape, you would actually then have more information to drive your decisions. This increases the chances that you're going to be able to find a really good clone from what you already have made, and then basically accelerates the library to lead selections. So this is our instrument. We've called it the LSA, and later on today, Dan, who's our field application scientist. Dan, can you stand up so everyone can see you? He is awesome too. He's going to tell you much more detail about it, but I'm just going to just give you a little overview so you can understand how it works. So integrated into this machine are two concepts, a single flow cell concept and a multi-channel flow cell concept. And these two microfluidic modules can be used to dock onto a chip. So allows you in the single channel mode to do what we call a lawning. So you can inject your sample and it attached to the entire surface or just flow over the entire surface. You could use this if you wanted to coat your chip, for example. And then in the other mode, you have 96 flow channels that are addressing the surface in a perpendicular fashion. The multi-channel mode is super efficient at getting sample to the surface because each channel has a back and forth sample delivery. So this allows a sample to have multiple opportunities to contact the surface. So it allows you to pull more stuff out of your sample. So a practical manifestation of this is if you had a very low concentration antibody supernatant, let's say like less than 1 micrograms per mil, you can actually enrich a large amount of that protein onto the surface much more efficiently than in something like a Biacore, where you have a single pass in a unidirectional fashion. So the printhead mode really allows you to array discrete spots in highly active form using flow printing. And then we nest this 4 times, and that creates a 384 array surface. And you can toggle between these 2 microfluidic modules. So once the printhead is docked onto the surface, this is a footprint of what the flow cells would look like. So each of these pink slits represents a reaction spot where you have captured or coupled a protein, let's say an antibody, onto the surface. And in between them, you don't lose any real estate on the chip for referencing, because we have these interspots shown as the blue rectangles. And as I mentioned, if the printhead docks 4 times in a nested fashion, you create this more crowded 384 array. And that's the principle on which our technology works. We usually work within a 384 format. Now, you don't have to derivatize all of the chip. If you wanted just to print 96 samples, you could come back to the chip at another time and address the other print locations. Once you have immobilized or captured your antibodies in the 384 fashion, you could then dock the single flow cell over the entire array, and this is an extremely efficient way of using your sample, like your analyte. One sample would then address the entire array. So this would be a high advantage over Biacore, for example, where you would need multiple analyte injections to address a much, much smaller number of samples. So this is what high-throughput kinetics looks like. So anyone who's doing SPR, probably having your mind blown right now, because something like Biacore 8K, 8 of these sensograms is the throughput per unit time. And here you have 384 sensograms, all generated at the same time. Each of these represents an interaction on a single spot. And I'll show you a little bit more detailed analysis. You can't really see these because there's so many of them and they're too small. But basically, you're not losing any quality with the throughput. So not only have we integrated flow printing with SPR detection, we've also integrated really powerful software with the hardware. So there's no point generating a lot of data if you can't analyze it and understand what it means. So this is an example of an output from our Kinetic tool. And again, Dan will go into more detail about how it works later. But we've incorporated threshold settings in there and quality controls so that automatically poorly behaving spots are flagged. And then you can sort the data in many different ways. And this makes it very, very quick to analyze your data set. So just a bit more up close and personal. So these are 12 spots from the array that I just showed you. And this is a single antibody that has been captured onto 12 discrete spots. And these aren't neighboring locations. These are different addresses across the array, kind of in a random fashion. And the antibody has been arrayed at 3 different densities, high, medium, and low. In this particular experiment, it's an anti-human capture surface created in a lawn method. And the point of this slide is really twofold. One is that it doesn't matter where in the array you put your samples. So it's independent of the geographical location. And secondly, it's really high quality data. So anyone who is familiar with looking at sensograms, here is a fit, a global fit with a simple Langmuir binding model, and that's in red. No funny business here. My former team will attest that I don't use complex models. I just keep it really, really simple, and you should be able to fit your data to a simple model most of the time. If you don't, screw the experiment up. So in red is the simulated fit, and in green and blue is the measured data. So the point here is that the simulated fit really describes the data very well, basically perfectly, because the lines are overlapping. And so you're able to get excellent quality data in a very high throughput. So let's say you didn't have 384 antibodies, because this paradigm of doing SPR on large numbers, hundreds of samples at one time, often is kind of beyond what people are institutionally set up to do. So oftentimes they'll only have like 40 antibodies to analyze. Well, you could fill out the capacity of the array with replicates. And this is an example of that. So each one of these spots has its own global fit, but then collectively you could get a mean and standard deviation and give some stats to the analysis. And this is super important when you're doing things like INDs or when you just want to get more statistical confidence in your numbers. So you can do this very, very quickly. So in a capture kinetics format, I just wanted to demonstrate both slow and rapid kinetics are well described on the LSA. And at the top here is 12 discrete spots of the same antibody, which is a high affinity binder. This is a picomolar binder. And the lower half is a very weak binder. There's multiple points to this slide. One is that once you've created the array and then you go to the single flow cell, you can titrate your antigen from low to high concentration. And so this allows you to really capture a very broad dynamic range in one single experiment with very, very little sample usage. So in this experiment, we started at, let's say, 1 micromolar and titrated down. So we're able, in the same experiment, to describe picomolar interactions and near micromolar interactions. And the highly parallel nature of the array gives you confidence that these are truly different interactions. So even a twofold difference between clones is resolvable. So Dan, again, will discuss this a little bit more later, but we have software that automates a lot of the kind of graphs and outputs that you folks probably like to present your data in. So here is an isoaffinity plot. If you're not familiar with it, you've got your kinetic rate constants, the association rate constant, and the dissociation rate constant on the two axes. And then the diagonal lines are isoaffinity lines. So anything on a particular diagonal line has the same affinity, but it's reached through different kinetic parameters. This is a really nice way of showing the kinetic distribution within your library. And here we've clustered the replicate clones for 3 different affinity antibodies, a high affinity, a medium affinity, and a low affinity, just to show you that the replicate spots cluster really tightly too. In our software, we have various QCs that I mentioned. It's always really annoyed me how software spits out biophysically kind of ridiculous numbers like 1 × 10⁻17, and then you'll report an attomolar affinity, which is ridiculous. So we've set it up so that you can put in limits. So on an off rate, for example, you can use the 5% rule, which is pretty conservative, and put something like a 1 × 10⁻5 as your KD limit. And that's why, as the arrow shows, that there are some clones that kind of cluster down a line because their off rates didn't show enough dissociation to resolve properly. So in our software, we just introduced this. I'm actually quite excited about this one. I really like this one. So say if you have replicate spots of a particular clone, the software will now automatically generate these histoplots. These used to take me a really long time to do, and usually make a mistake doing it too. But the point here is that these are really tight groups. So yeah, it's a very highly reproducible technique. And then, these help you with your presentations as well. So this would be an on rate, for example. And this represents 34 unique clones spotted onto 8 or 12 different places around the array. This would be the off rate, and then the overall affinity as well. So you're probably all thinking, well, that looks amazing, but how does it compare to Biacore? Because Biacore's been around forever, and it's a very well-established and trusted technique. So we took this challenge head-on, and we're in a collaboration with Adimab. They provided the set of antibodies that I just described to you, and they ran their antibodies on their Biacore 8K in a single run, because it actually used a lot of antigen. And I ran the exact same experiment. We ran it in-house on different types of chips, and we ran these 30 or so clones in replicate. So on our end, we have statistical significance. We've got error bars on the y-axis. They don't have any error bars because each KD value was an n of 1. So you can see with your eye that it's an amazing correlation. It's not just a good correlation. It's basically identity. If you did an experiment twice on your Biacore and you got a 1.2 or 1.3 fold difference between the numbers, you'd say you basically got the exact same answer. So again, the point of this slide, it's actually got a lot of different points. One is that if you match the chip types, in this particular case we use flat chips, which are actually more suitable for antibody kinetics than 3D hydrogels. These actually match better the affinities that you would get from Kinexa or MSD, some kind of solution method. You can get really excellent agreement with what you would get on your Biacore across a really broad dynamic range. Here I've shown 4 orders of magnitude. It could have been even broader if the affinities of the panel that we were looking at happened to be broader, but this was the affinities of the panel. And there's 36 different clones here, so it's not like we're only having like a sweet spot where we work. It's pretty amazing. And just bear in mind that we're doing this in a 384 format, so we're 50 times faster than a Biacore 8K. And because once you've created your array, you're titrating the sample over the entire array. You literally make 8 tubes. You're 1 micromolar, and then dilute down. And so you're actually using only like 1% of the sample that you would use on a Biacore. So we use like a few micrograms for this experiment, whereas our collaborators used milligrams. So you're able then, in a single day's run, to get full kinetic profiles in a high-resolution manner on 384 clones. They could have been different clones. For the purpose of this experiment, they were replicates of a smaller panel. And I just showed you that we have really nice batch mode fitting in our Kinetics software to enable you to get to the answers just really, really quickly. So I'm going to switch gears now to have a little bit more time.
You're doing great.
Awesome. So I'm going to just tell you why it's all about the epitope. So you guys probably know all this stuff, so you can just tell me kind of to move along if you know all this stuff. But for those of you who don't know, epitope is really, really important when you're characterizing antibodies. It's one of the 2 main parameters that you look at when you're looking at binding properties. One is the kinetics and affinities that we just talked about, and that tells you how strong the interaction is. And the other is the epitope. It tells you where on the antigen you're binding. It's kind of like if you think about the world, think about the Earth as being your antigen. Where your antibodies bind are like countries or spots on it. So you might have an antibody that binds in Paris, one in Singapore, one in wherever. And antibodies that bind in a similar spot oftentimes have a very similar function. And so epitope is a really, really important characteristic because it's actually the only parameter about an antibody that you can't engineer on purpose. So I like to think of it as an antibody's like born with a particular specificity. All the other parameters like affinity, you can mature with engineering. So the paradigm for epitope is that you have to select the right epitope. The other parameters you can actually engineer or optimize. So you can't, right now it's impossible in silico to design an antibody with a particular specificity. It just can't be done. And you can't predict if you have a sequence of an antibody, you can't predict where its epitope is. There's tons and tons of papers on in silico work, and it just doesn't work. We're very, very far away. And this has manifestations like for ADA, that we don't understand ADA, we don't really understand B-cell epitope responses. So until we do, you have to describe and characterize epitopes empirically. In the drug discovery industry, most people are working on the same targets. So you have to be able to differentiate your antibody from someone else's. So just giving the example like PD-1, there's over 40 PD-1 patented antibodies. There's more than 20 in the clinic. There's 5 approved, I think. And what makes them different from one another? Well, epitope can be a characteristic that can give you a little bit of an IP advantage. And when you're starting out a project, it's really helpful to know where your epitope lies in the epitope landscape of that target. So if you had the ability at the very early stages of research to understand your epitope landscape, it just gives you just more information to direct your research. And it allows you then to select antibodies with functionally relevant epitopes, and it allows you to prioritize your resources better. So Array-SPR is uniquely suited to help you characterize epitopes, and I'm going to show you some binning stuff. So I've been working on binning for many, many years, and I'm really excited about array-based high-throughput epitope binning. So many years ago, I thought I was like a rock star doing like an 11 by 11. It took me 2 days on a proteon, but that was actually quite a large matrix back in the day. And then I wrote a paper, I think in 2012, doing a 36 by 36. I was like, oh yeah, this is so cool. But with our current system, we have like 10 times, well, more than 10 times, because it geometrically scales. So you can now do a 384 by 384 as standard. You just like think about this, it's like pretty insane. This is like 150,000 binding interactions that you can do walk away. And the really cool thing about array-based binning is once you've created your 384 array and you go to the single flow cell, you only need one aliquot of your antibody as the sandwiching partner to address the entire array. So your antibody consumption does not scale with the size of the panel. If you're going to do a large matrix on an octet, the amount of sample that you would need would scale with the number of antibodies that you were investigating, and it becomes ridiculous, and you basically just don't do it. So I want to just draw your attention to the pictures here. So on the right is a heat map and a community plot that we published back in 2016. So back in 2016, we were able to demonstrate the 384 array-based binning using the combo that I spoke about before with the CFM and the IBIS. But now this is an automated assay on the LSA. In the same year, a very well-respected group published a very regularly sized matrix that most people are doing when they're not using SPR in an array-based format. So this was an output from an octet, so a 13 by 13. So I like to make the analogy here is like having black and white TV versus high definition or whatever the new like modern TV is. But you basically have more resolution when you have more pixels, more probes within your library. You can pick out these tiny nuances that can actually make the difference between your epitope and a competitor epitope. So because our throughput is so huge relative to any other biophysical tool, we really are transforming the binning paradigm. We're enabling people to investigate and interrogate epitope very, very early in the drug discovery process. So this then changes your workflow because you have information that you just didn't have before. Kind of like to make the analogy here with sequencing. back in the day, I did a manual, Sanger sequencing using a gel, but now you just kind of send it off, and the next day you've got your sequences back. And knowing the sequences allows you to prioritize your clone selections. What if you could know your epitope landscape in as facile a manner early in the drug discovery process? So here I've just kind of lined up the LSA and the Biacore 8K and the octet HTX, and we're just looking at antibody consumption if you were going to do a 384 by 384. So in our machine, you're only using about 5 micrograms per antibody. With the Biacore, you're using about a mg, and also with the octet you are. Now, a mg is a lot of antibody, but more importantly, one, you probably don't have a mg of 384 antibodies. Two, it's a complete nightmare to plate all of them out. You basically wouldn't do it. So people just don't do large-scale binnings on other instruments. So you're basically missing this information. So this is a collaboration. Actually, Katie here from Ligand will elaborate more on our collaboration, but this is something that we published last year. And this was only a 192 array-based binning. But this just shows you the clarity that you can get in large scale when you do binnings. Another thing that our customers really like is that you can also show the results of a heat map in terms of a network blocking plot. So our CEO likes to call these the Facebook of antibodies. It basically shows you the clusters, which antibodies are hanging out with which other antibodies. These different clusters show you the epitope landscape. You can add in orthogonal data. So in these examples, we've colored them by the subdomain of the protein, the library. So say if you have 2 different libraries, a wild-type chicken and a transgenic chicken, and you want to see what their epitope landscape is like, is it same or different, then you can color the plots accordingly. So this allows you multiple parameters viewed with an epitope-centric focus. And then the bottom panel shows you mouse cross-reactivity, for example. So you can very quickly navigate your way through the panel. And I didn't mention it, but the name of our technology, the LSA, stands for Lodestar Array. We like to think of it as a star kind of guiding you through your antibody discovery process. So in summary, high throughput SPR is a disruptive technology, and it now shifts SPR from its traditional role as a secondary tool to a primary screening tool. So it expands the use of SPR, basically. Screening and characterization are basically the same step now, so you don't have to screen in a high throughput but low information content way and then later on get your detailed kinetics. You can get your detailed kinetics right out the gate. So this allows very detailed information of all of the clones early on and helps you decision making. And relative to Biacore, I showed you the data. The data speaks volumes. You're getting the same quality. You're getting the exact same kinetic rate constants as what you would get from your Biacore. Less time, 50 times faster, 1% sample.